As a group of undergraduate students, we are initially interested in the stress levels that arise during a school year. But as we got more involved in the work that Dr. Nazad's lab was doing, we found how cortisol levels can be major identifiers in other illnesses and conditions. These can ultimately be used as a precursor for condition identification.
So what we have developed is a microfluidic chip that has the ability to produce quantifiable results of cortisol levels within saliva. Now we have a simple task of taking a salivary sample from a patient, and the chip is able to process it within 10 minutes.
Now in the lab, we have been testing for cortisol levels through saliva samples from individuals. It's a challenging way to test for cortisol, but we believe that this type of point-of-care testing is very necessary, and also the way of the future for medical diagnostics.
The challenge for us has been to fabricate and optimize the design of the microfluidic chip using COMSOL multiphysics and AutoCAD, so that the chip is able to perform the way we want it to, while still retaining the function of being able to be assembled autonomously using the Mechademic robot. Once the salivary sample is placed in the inlet well, capillary action initiates drawing up the sample, splitting, having some of the sample travel to the redox chamber and some traveling to the mixing chamber, where the available proteins will attach to the antibodies. Along with capillary action and the wicking rates of the zigzag channel, the sample will eventually end up on the right side in the absorbent pad.
To achieve semi-scalable automation of the microfluidic biosensor assays, we're using the Mechademic Mecha 500 robot, which is consistently accurate to within 5 micrometers. Together with a pneumatic module and the robot's 6 degrees of freedom, we're able to freely pick, place, and peel the layers to achieve the automation flow we desire. We wrote a script in Python that runs on a Raspberry Pi and sends commands to the robot and the solenoid valves.
The robot utilizes a 3D printed suction cup which is attached to small diameter pipes to create a suction area to help pick up each layer.
The microfluidic biosensor assay is a device that uses tiny channels and chambers to perform complex biochemical reactions on a small scale. When a biological sample is introduced into the assay, it reacts with various chemicals and produces a signal that is detected by the electrode sensor. Our system utilizes an electrode sensor connected to a Raspberry Pi Zero via a GPIO port, which acts as a powerful computing platform for analyzing the data. The electrode sensor is a cutting-edge technology that allows for precise measurement of cortisol levels in biological samples.
The work that Dr. Nazad has produced so far in his lab is very impressive. They initially found that there is a very good correlation between stress levels and saliva cortisol levels. So going forward, we are very excited to be continuing the progression of this research to form new breakthroughs.